want this breakfast seminar where we will discuss the usefulness of a whole body monitoring system with bioluminescence. I think this is a hard word to pronounce, but for in vivo, ex vivo, and in vitro applications. And this system is expected to support drug development in preclinical environments by following disease progression or efficiency of treatment in preclinical research. During the past years, RISE have used the camera in collaboration with the Swedish biotech company Anocca. And during this hour until 9 o'clock, we will listen to Hugh, who is CSO at Anocca. And followed by him, we will meet Evelina Lindmark. So she's a researcher and study director for in vivo studies at RISE. And then we will also meet Matilda Bäckberg, who is a project manager for Pharma Office and director for RISE Laboratory Animal Operations. And finally, myself, who has the honor to moderate this morning. And my name is Cecilia Hyrén, and I'm a research and business developer at RISE. But before we start, I want to mention that between each speaker, we will have time for questions. So please write your questions in the chat or shout it out during the question time, and we will do our very best to answer it. But first of all, the project manager for Pharma Office and also the director for RISE Laboratory Animal Operation, Matilda Bäckberg. Matilda, could you please tell us a little bit about Pharma Office at RISE?
Just shortly. Pharma Office at RISE has been a project for 3 years now, and it's kind of organizing the overall drug development offerings that we have within RISE. It's a kind of gateway to reach the experts or the speaking partner that you need in different steps. Mostly in the preclinical parts, but also we could be helpful when it comes to the clinical steps as well. So from Office at RISE is intention is to make it a bit easier to reach out for our customers and partners and give them the expertise they require.
Thank you. And this is actually the second breakfast seminar. The first one was held in October, and there we focused on AI and drug development. How did your experience the first seminar?
It was the first one. It's always an experience when you do it the first time. But then, as you said, we had the focus on AI, and it's a quite and within the drug development, it's kind of broad focus. We try to see if we could get into the drug development process in different aspects. So that was a quite wide one, and now we're more direct, working with a more specific instrument in the preclinical work. So we try different things and see how it turns out and how the interest is in. And but just for those who didn't listen the last time, we recorded at that time as well, and you can find it on the Pharma Office website. And then the keynote speaker was Eric Illipe, and he is one of many AI researchers at RISE, and he has a focus when it comes to natural science, and his skills becomes very good when we develop systems within chemical route scoutings and safety assessment, for instance. So if you're interested, you can still listen at that one.
Yeah, it was an interesting seminar. I agree, actually. But to this morning we have a complete different theme. so it will be really interesting. But can we expect Pharma Office Breakfast Seminar to be a series of seminar from now on, or what do you think about that, Matilda?
Oh, yeah, that would be good, wouldn't it? Let's hope so. At least it's the intention. Maybe 2 to 4 times a year. It depends on the interest and the time we have.
Yes, yes. Thank you, Matilda. And you will come back in the end of this seminar and talk about preclinical research at the Pharma Office at RISE, and also the collaboration with Anocca. Thank you. And our first speaker is actually the CSO at Anocca, the company behind the monitoring system. So welcome, Hugh Salter. Will you start? Just tell us a little bit about what you will speak about in your presentation. You have muted, Hugh. We can't hear you.
Apologies. Can you see my slides and hear me?
Yes.
Okay, great. Yeah, so what I'm going to do is actually tell you a little bit about Anocca, about the technology platform that we've developed, and why the collaboration that we set up with RISE is important to us. And I'll end with showing a snapshot of data, and then Evelina can explain how that data comes to be. Okay. So just first, a tiny little bit about Anocca, biotech company, grown to around 90 people over the last 8 years. The work I'm going to talk about now is something that we really started concentrating on to the exclusion of other stuff, everything else, around 2007 to 2016, 2017. So what you're seeing is a result of getting on for 5 years work of developing a platform. And the specific point of what the platform does is to develop a platform for the biology of understanding T-cell biology, and the output of that platform is TCR receptors that have the correct characteristics for being used in clinical studies as products. We're moving towards proof of concept clinical trials, so we want to start in 2024. And an obvious bridge between those two points is a translational platform to evaluate efficacy in animal models, and that's the context. Physically, we're actually in the same building as Evelina and the team. We have labs on the 2 floors above the RISE facility, and we also, on the same site in Södertälje, have a manufacturing facility which is in a different building. So we feel we're very well equipped physically, and the proximity to RISE has been a great benefit to the collaboration. We can literally just walk down 1 floor of stairs. What I wanted to start with, without making anyone's lives difficult at this time in the morning, is just to say a very, very few words about the type of biology that we're trying to understand here. The type of the biology is the adaptive T-cell response, typically in class 1, so the CD8 type of T-cells, between an antigen-presenting cell, which shows a target on epitope, an antigen, whatever you want to call it, of interest in the context of a specific HLA molecule. And this of itself is a complex piece of biology, because there's polymorphism, diversity, and therefore segmentation of patients on the HLA side. But an even more complex part of the biology is the fact that the repertoire of potential T-cell receptors that can recognize these HLA-restricted presented epitopes canonically on the surface of a tumor cell or a virus-infected cell are incredibly diverse. There are, because of the recombination biology that sits behind them, and this has led us to develop a suite of tools to analytically understand and develop TCRs. And I'll say, just to loop back to the introduction, that this is not an area of biology that's particularly amenable to AI at this point in time, and that's simply because of the lack of parameterization around and sheer complexity, in particular the interaction between a T cell receptor and the HLA restricted antigen. So the way in which we've chosen to build our platform is to build a functional platform that recapitulates in cells the biology you see in the top left, an antigen presenting cell showing a complex range of antigen HLA presentation on its surface, once again canonically a tumor cell, and a T cell that's responding. And the way we address this is to make families of what we call engineered programmable cells that recapitulate both or either of the antigen presenting aspect. So these are cells from which we've removed the native HLA and TCR components, replaced them with genomic cassettes into which we can flip in genes of interest, making monoallelic cells that present antigens very specifically, that become therefore target cells, if you like, in the context of an experiment. And we can, on the T cell side, similarly make cells where we remove the native TCR and HLA components and we replace them with TCRs of interest. From an analytical point of view, the key point here is that we simplify the system, we make it into a cellular response assay that we can hook up with various promoters and bits of technology that we don't need to talk about today, but allows us to, in particular, to study this interaction between the antigen and the TCR in an extremely effective way. What we do then is build these assays together in various ways to allow us to map the antigens that are found that are relevant for particular targets, to isolate TCRs for them, to validate them as targets, building on the validity of the relationship between a specific set of TCRs and their cognate antigens, and then a big triage, which perhaps a story for another day in discussion with an organization like RISE, about how we use these assays to front load safety, noting that TCRs have an inbuilt potential risk, in particular for off-target cross-reactivity reactions that needs to be tightly controlled. What comes out of this process is a set of T cell receptors that are candidates for translational and clinical experiments. So the therapeutic concept then is that we're going to take a T cell, which some people call an effector cell or a killing cell, and we're going to introduce into then a genuine, authentic primary T cell, typically derived from the patient themselves, the TCR candidate of interest, and then in vivo we expect that to target a tumor cell that we have shown and selected the patients that specifically express the combination of epitope and HLA to which the TCR is targeted. So the product is patient's own autologous CD8 cells that have been transduced with one of a set of specific therapeutic TCRs. The way this works in the clinic is that the patient is leukapheresed, isolates CD8 cells. We introduce the candidate clinical TCR, typically by transduction with a lentivirus. We expand, purify, and then in the clinical setting would infuse those back into the patient. In the preclinical setting, these cells become test objects, test products that can be assayed in a range of assays for their translational properties. And at present, as I just mentioned, this is nearly always done with autologous patient's own cells, but in the future we'll use different types of effector cells that can be sourced from, for example, donor cells that have been gene edited, or potentially even stem cells. To build a translational platform around here, we've developed a platform that utilizes both the target antigen presenting cells that we have, but also cells that are derived from other sources, whether they're standard immortalized cancer cell lines, patient-derived cells, or, for example, organoids, but primary material. And part of what we're doing then is building a platform that allows us to have available a range of techniques to allow us to assess these product cells, which are capable of killing a cell presenting the HLA restricted target in a translational framework. And here then is just a tiny snapshot of data that of how these kind of experiments work. On the left, an in vitro assay showing increased amounts of killing that is specific and selective for the target by these therapeutic cells expressing a candidate TCR, selective for the target and selective for the HLA. On the right, a snapshot of data showing how an example of how these kind of experiments work in vivo. The axes are flipped, which I realize now, in the sense that increasing axis on this right hand side in vivo shows increasing tumor burden, the tumor burden being measured with the bioluminescence probe that Evelina will talk about. And you can see in this case that treated mice at 2 different doses stabilize and don't tolerate tumor growth, whereas the untreated mice do. And the target here then is a cell line representing a target positive antigen and HLA expressing cell. There is a disease mimic into which we've also included luciferase's reporter, which I'm sure Evelina will mention, and those have been engrafted into immunodeficient mice, NSG mice, and then treated with our candidate effector cells. The immunodeficient mouse component is important, of course, because these are human cells, and they need to go into an immunodeficient mouse, or the mouse immune system will eradicate the target cells. So that was my very brief introduction to the concept of TCR presenting effector cells, and antigen presenting target cells as a test system which can be introduced into both an in vitro system, but also into an in vivo scaffold for analyzing and understanding the potential efficacy of these products. So back to the group. And you're on mute, Cecilia, now, not just me who screws up.
I said, thank you so much, Hugh, really interesting. And we got a question or a hand. Anna-Lena, please.
Yes, hello. My name is Anna-Lena Spets. So very nice talk and very interesting platform. So I have a brief question. So now you show data on the immunodeficient mice. So I'm wondering if you've done experiments also in using other than tumors and models, having murine cells, murine T cells, engineered T cells, and murine tumor cells. So my question is this: can you say something about the duration of the effector functions of the engineered T cells? In other words, will they start to upregulate CTLA-4, or will other breaks on the immune system will put on, or perhaps you can overcome that by multiple injections? But you get my point. I'm wondering about the duration.
Yeah, in animal models, the duration of the effect doesn't normally last longer than the mice do, if you know what I mean. The activated cells, after they've killed, will return to relatively small levels. In the human in vivo setting, there are ways to think of combination therapies in various ways, which I can't go into, to ensure that they remain engrafted in the memory compartment. The cells that we produce have been polarized as memory cells to start with, I should say, by the manufacturing. So the durability is related then to the phenotype of the cells, the level of tumor response, the type of tumor response, the tumor microenvironment, the level of CD4 support, helper support that either occurs or is induced, the level of epitope spreading. So it's very difficult, and this is why animal experiments are helpful here, but the genuine experiment is one that you do in humans. You can do, of course, as you suggest, mouse TCRs into mouse tumors, but that doesn't recapitulate the human system, either from an HLA perspective or a TCR perspective. So that is a fundamental limitation of using rodents to translate human cells in this context. And I think that challenge is, for example, recognized by the regulators.
Okay, yes. Okay, yes, thank you. That's very interesting. Thank you.
Thank you. And we have another question, Anna Ridderstad Wollberg.
So, hello, you. I'm wondering if I understand correctly that the drug product, those are the autologous CD8 T cells, and that they are not monoclonal, so it's polyclonal group of cells that have been transfected with the receptor. But have you thought of the definition of what is the final product for your quality assurance, the phenotype of the cells, and how will that work?
I'm getting close to revealing some secrets here, so maybe I shouldn't.
That's what happens when you have immunologists in the
yeah. So there are specific release criteria associated with the product in terms of its phenotype and genetic load that may or may not be associated with the process, the production process itself. And these are, I'll just say that these are well understood parameters.
So, because I guess you haven't stripped everything on the cells, so you have a definition already set from the beginning. Yeah,
indeed. Yeah.
Thank you, Hugh. And we can come back. We will have a group discussion later on. But I think it's time to go further in the agenda today, or the program. So now it's time for Evelina Lindmark, the researcher and also study director for in vivo studies at RISE. She will give us a presentation about the monitoring system. So welcome, Evelina Lindmark.
Thank you. So I will share my screen. Yeah. And there we go. Do you see it well?
Perfect.
Yeah, great. Yeah, so I will talk about the actual in vivo system, the camera we use, and how we use it, and how we work with our animals at RISE. And so here is the actual system. It's Newton 7 is the name of the instrument, and it's purchased from Wilbur, who makes it themselves, and actually the lens as well. And to be able to pick up the bioluminescence, it has a CCD sensor that will pick up luciferase, the luciferase signal that is what we transfect, the cells with, or you can conjugate that to basically anything. And you will have to use luciferin. So luciferase is an enzyme that will oxidize luciferin. So when it comes into contact, you will get a chemical reaction which will generate the light, the actual bioluminescence that the sensor will pick up. And the system has a lens with aperture that is has a very low f-stop number, and the lower the number, the more light can go through the lens. which means it can pick up very faint signals of bioluminescence. And if you would have in a petri dish in there with cells, it will be able to pick up a single cell in there. And to the right, you see the actual inside of the camera, and you see that stage where you have the anesthesia masks. So it's for up to 5 animals. So the field of view will be 20 × 20 centimeter maximum, but you can adjust that. So it's possible to use up to 5 mice at a time and 3 rats. And I also heard that there are groups that have been using hamster and a machine as well. And this stage the animal lies on, it's possible to slide it. You slide it towards you to actually, to place the animals in there. In the heated stage, well, for standardized operations, it will reach 37 degrees. But that you can switch off if you have in vitro applications to use it, and you can decide the actual heat on that. So with the bioluminescence imaging system, you can do a variety of things. So we can do the tumor monitoring, as you talked about, which I will go into in a bit more detail. Next slides. You can also do tracking of cell migration. So if you have luciferates inside of your cells, you can use to track them. For example, you can check where the immune cells home, to which organs they go to, for example. And you can also use it for biodistribution of drug molecules and nanoparticles. Basically, everything you can tag with luciferase, for example. And there are a wide range of different types of luciferase commercially available. We've been using the luciferase called Firefly, mainly. I think that's the widely used ones, but you have the vanilla and the gauchia and different types of them. And we also have the ability to conjugate if you don't find your right commercial product. We have the ability to actually do that type of conjugation at RISE also. And you can also use the bioluminescence to visualize vasculature and microcirculation inside the animals over time. And to the right bottom, we have a picture of the 3D tomography you can use for the cameras. You will take pictures from 3 angles, and the program will manage that into 3D picture where you can apply the organs, the skeleton of the mice. So it will adjust that to the actual size you have on your mice inside the camera, and then you can use that to localize, for example, your tumors to an organ and see where it's situated inside of the animal. And I forgot to mention on the top right is a picture of tumors that has been implanted intravenously inside of a mice, and that's approximately after 20 days to the left in the same animal when the tumor are growing inside of the mice to the right. And then you can also use it for in vitro. You can put your petri dish inside of the camera as well. And you can take out, towards the end of a study, you can take out the organs and image them separately as well, if you like to look at the actual signal from a single organ. That's possible. Yeah, so now I will tell you how the tumor studies we run with Anocca, how we work with the animals during these studies. And we usually get the animals from our vendors 2 weeks before we start the study. And during that week, the animals will acclimatize and will be trained in the procedures that we will perform and they will get used to us and the handling, but we will handle them with our hands. And typically the training sessions takes place at 5 occasions with 2 minutes for each animal. And for example, in these studies, we use caliper measurements. We do weighing of the animals. They do get IV injections, subcutaneous injections, and they get sedated. So they will be subjected to this handling. So for example, if you do IV injection, you will touch the tail during the training, or the scruff if you do the subcutaneous injections. And I put in, yeah, it's supposed to be a link in here, but if you are interested to see how we handle our animals, there are a couple of movies. So if you go to ri.sc, you can search on 3R and you will find that page where we have a couple of short movies and a long movie showing all the procedures we perform and how we do that without restraining the animals as much as possible. And yeah, let's go ahead and check those out because they're really nice, and it's really, really de-stressing for the animal technicians that work with the animals, and also the animals too, that everybody is calm and you don't cause unnecessary stress to the animals. So after that period, we usually start by injecting the luciferase transfected tumor cells that Anocca comes down from 1 floor above. And we inject those in the animals, and then 1 week later, they are subjected to the first session in the camera. And so to be able to actually visualize the light, as I told you about the luciferin, the substrate for luciferase, we do inject that before we sedate the animals and before they go into the camera. And before we started, we needed to take kinetic curves to see after what time we have the optimal signal, and for the duration of that signal to be stable. So you need to do that timing before you start. And so typically we wait, because we gave the luciferase subcutaneously, so we wait for a period before they go into the camera. Yeah, so the actual sedation of the animals, so typically we use 5 animals per group. So they need to be very closely injected to have them very like an even situation in the footage. And they need to go down at the same time as well. So normally we do want to sedate the animal 1 at a time to make it a bit more calm for them. But for this project, we needed to use the box for them to go down simultaneously. And then in the left picture, to the right, you see the actual camera on the outside. Maybe I didn't show a picture of that previously. So the tricky part then is to get them in there and switch on the masks inside of the camera without the animals waking up. And yes, exactly. They have been transferred then to the animal stage. And then after that, we do sort the animals before we put them inside of the camera to have them in the right order, even though you do see the tail markings in the pictures. But we make sure they are in the right series. And then they get eye drops to not drying out the eyes during the sedation period when they are down. Then you have to adjust the position a bit of the animals. And then you close the door and go over to getting the focus on the actual animal image that will be overlaid with the signal from the luciferase. And after that, you do the acquisition. You maybe have to adjust exposure, and the camera typically you can set that however you want, but you can do that for an automated setting where the camera will see where the optimal exposure will be. So it's basically a click to get the right exposure. You might have to adjust that, and might have to adjust some of the animals depending on how the tumors are situated. And then you capture the image, and that's basically it. Very simple procedure. And then we transfer the animals to wake up in their home cages, and then we upload our image, and Anocca takes care of the image analysis for this study. And this is an example of how the software analysis looks. So after this first week, we usually do this once or twice a week. And these studies usually go on for 2 months. And in some of the studies, we've been doing treatments, some we've been setting up the actual tumor model. So we do treatment where the TCR, the T cells, are being injected as well, typically IV. So that will be the middle of the studies. We have been taking some blood as well for some of the studies. And as I mentioned previously, we do the caliper measurements for the subcutaneously implanted tumors to have a comparison of the data. Yes, that was everything to tell you. So please, if you have any questions, just go ahead.
Yes, do we have any questions, comments for Evelina? Thank you so much, Evelina. It was really interesting. Do you have the link maybe to the movies that you can post in the chat? It would be great.
Yeah, that's a good idea. I could
You
put
get
it there.
hands, yeah.
Evelina, how handy is it, the instrument or the camera? How many people are you around when you do the imaging?
Yeah, so we are typically 2 persons doing that. So one person takes care of the actual footage, and one person takes care of the injection with the luciferin. And that can be varied in between people as well. So it's a varied study where you get to do a lot of different things. And the machine is very easy to handle. So the choice was Anocca's to get that camera, but I'm really happy for that because it's very easy to use. And it's good that we have several people that can use it, and it's easy to get people trained on the machine. So that's quick, so that we can have a lot of resources around that. And that makes us a bit more flexible, I think, as well.
Great.
You said something about the necropsy, and you look at the specific organs. That's a possibility as well. Do you need to work very quickly then? And
No, so it's a matter of when you bathe them in luciferin, because they won't light up until you do that. But as long as you usually should do that quite quick, of course, because the organs will go down once they're outside of the body. But yeah, as soon as possible. But that I haven't tried myself yet.
Thank you, Evelina. This was a great insight for me, at least, to just see how you work with that. Is it any more comment or question to Evelina before we start the discussion? You can come back with questions later on, of course. So now we will have a discussion of future opportunities within preclinical research. So I wonder, because this monitoring system is a fairly new type of technology, maybe this question is to you, if you can give any example of how RISE helped you at Anocca to develop and also improve the system, because you seem to have a really tight collaboration.
I think there's been a challenge in taking a whole bunch of papers that describe how this has been set up with different technologies and not different type of mice. They're usually this pretty similar mice, although the NSG have become an established standard more or less. But different types of cells. And I think there's an interplay there between, you know, how the cells have been created, the animal procedures themselves, and for example, how the injection, how a subcutaneous injection, for example, affects the size of the tumor and the potential for it to spread, the range of behaviors of different types of cell lines, the type of, you know, cytokine support that may or may not be needed to keep T cells proliferating in this immunodeficient environment. And I think that's been something that we've collaborated tightly with Evelina on as we set this up, so that it becomes a more and more routine model. So I think the physical proximity actually of the two organizations, you know, literally 20 steps away from each other, has definitely helped in this.
Evelina, would you like to comment?
I mean, in general, it's a very fun project, I would say, because yeah, since we have such close contact and we are able to kind of see the progress in real time, basically as well. And I think my colleagues really, yeah, do like our collaboration as well. And it's very easy, and I think you have been very good at adjusting to how we work with the animals as well, and take that into play.
Yeah, for which I take no credit whatsoever. That's all Hannes and his team.
Yeah, I know. So it's really fun and easy collaboration. And I think we're doing a lot of use together to move this forward.
I think it's a great way to develop new technology, which are beneficial for us really in the future. But how can the customer benefit from this technology? Customers from RISE? Do you have any examples?
So like what I did present then was the different types of techniques where you can use bioluminescence camera. So I know it's usually widely used, or the main use is probably in tumor studies, but I think science is going a long way with using it for other applications as well, and also when it comes to what you can actually tag with bioluminescence. And I know that this camera, you can also add fluorescence module to it as well, so that will give you a bit more flexibility as well. That is a possibility. But I think since the luciferase is so widely used, you can use it in so many different applications. And I think we are looking into looking at the biodistribution as well of drugs, since we do toxicology mainly. And yeah, that can be an interesting way to go as well, to look more into that.
Yeah, what should you say? Yeah, sorry, we have a question.
I have a question to hi Hugh. Been listening in the corner here, you didn't see, but thanks for the presentation, both of you. As you just mentioned the word toxicology and safety, do we see any way in which this technology can actually address potential safety paradigms with this kind of therapy? Obviously these mice and rats, they're not humans, and obviously you have, your cells are human based. So, but is there any way, do you think, that a technology like this could be used to understand if your engineered cells are, for instance, accumulating in areas you don't want to have them, accumulating in inadvertently. I'm saying, you know, lymph node colonization, for instance. I'm thinking out the top of my head, but obviously the technology, being able to see things where they are is important, I think, from a safety perspective as well. Maybe in the beginning, not at the end for regulatory purposes, but trying to understand that.
I think this, let's say the current the type of product we have now, as I mentioned earlier, in response to Anna's question, it has a very defined, well understood phenotype, a very well understood response, for example, to cytokine stimulation in both in humans and in immunodeficient mice. So you don't, I'm afraid to say, you don't learn very much about safety from these. This is primarily an efficacy. The safety liability associated with these products falls into kind of two buckets. Bucket number one is the issues that are inherent to the T cell itself, the TCR receptor itself. And those are canonically handled with the kind of platform that, well, not canonically, best handled with the kind of platform that we've developed. And then the other issue is actually more around clinical safety, which is, it's not really a result of the of these types of T cells per se. There's an interplay between the kind of regimes that are used for lymphodepleting the patient and the often hematological consequences of that, the consequences of high dose cytokine support, again, in the patients, and in the field of cell therapies of these types of T cell therapies in general, more problems that are associated with CAR T products than TCR products, in particular cytokine release syndrome. And this is, again, because of the need for an intact immune system to look at this. This is very difficult to translate from a rodent model into humans. Yeah, yeah. So, I mean, what we've tried to do here is essentially make a product that takes with it as little of that potential liability as possible by being as close to a native cell as we can possibly get them.
Okay.
Right? Yeah.
I got you. Thanks, Hugh.
Yeah.
Cheers, mate.
I'm curious about you, if you could tell just what do you see as the next step in this technology and for Anocca?
I think that's not technology. The way the field is going, and this is where I was going to add something to the answer to Ian, where these may be useful, these animal models may be useful, is that in the future the source of cells will be allogeneic, not autologous, and as I'm sure many of you know, there's huge amounts of investment around the world, and some work going on in this country as well, of course, about how that will work. And also lots of people taking different types of effector cells, putting different types of modulations in there, immune shielding, decoys, tumor microenvironment modulators, all sorts of weird and wacky things. I think some of those will have consequences for how this sort of scaffolded interaction between a target cell and an effector cell can be monitored in a mouse system, I'm sure. So I think that's where this technology may have a role in the future.
Great to hear. Did we have some more comment or question? Maybe. Any more? Look, we have two experts here. So take the possibility to ask all your questions. Okay. Do you have something more, Evelina, or you, that you would like to mention in this seminar?
I just want to thank Evelina and the team at RISE, and also take the opportunity to thank Hannes and his team at Anocca, who've been the main people interacting with the RISE team for a great collaboration, and look forward to it carrying on.
Yeah,
Nice.
likewise. And yeah, I would like to thank my team as well, because we didn't have this equipment, because Anocca placed it at our place, but they've been picking up very quickly and really been eager to learn the machine. So it's been really fun. And as I said previously, it's really fun to work with Anocca as well. We really are part of that work, and we have close interactions, and it's really nice to follow everything in real time. So we're really happy with our collaboration.
Perfect. And I wish you good luck in the future, and maybe we can come back in a few years and just study your collaboration again and see what happened, because it happened a lot.
Welcome.
So yes, thank you so much. And I would like to ask Matilda, as we said previously in this seminar, we said that you should come back to us and talk a little bit more about preclinical research and also pharma office at RISE. I actually have a question, how pharma office at RISE differ from the offering of commercial player or private actor? Do you have any answer?
Well, first of all, RISE is a state-owned company, and we exist because the Swedish government has given us a social mission to work for a sustainable growth of Sweden by strengthening the competitiveness and renewal of the business sector and contributing to the innovative development of Sweden. So, and just focus on what we are doing in the pharma office at RISE. What it means for that, it's that we strengthen the Swedish pharmaceutical companies, and by supporting the development and processes in various ways. And the good part about RISE is that we can offer research and development assignments with different forms of funding models. For example, fee for service. We perform applied research from commercial actors or by research grants. We could be a partner in a joint exploratory research project as well. So there are opportunities. And just to be clear about it, we are what's this called in English? Aktiebolag, AB. But we are on an open market, but we are a purpose-driven organization, and we are on the market just to create the best conditions for our SMEs and make them do the best on an international market in the end. And the benefit of our work is just as in this way, continuous development of activities, provide the support that the companies Anocca needs, and to know what to do and to focus on, we need to get the information from the pharma sector. Where are the needs? So it's a close interaction with the SMEs. That's very important. So just look at the way that we work with Anocca. We described it several times, but as we heard, Anocca, they have the lab space and they have skilled researchers already within the company in their team. But as you heard, we are situated very close to each other in the same building, and we met up, and new ideas grow, what we could do together. And as RISE is like, we are like Barba Papa, and we move around and turn out to fit in where we are needed, and and this way we could be kind of extended arm of their R&D unit, and could give Anocca another scientific aspect of their ideas. And in this way, our machine park at our facilities at RISE got the new. They moved in this whole body bioluminescence imaging system. So that's what's the solution this time. We offered space for the camera, and we offered our animal technician and researchers to move forward in their drug development process.
Yes. And you have a lot of researcher in-house, and you mentioned AI, like one on the expertise and also this monitoring system, but it's many more.
Yes, and that's the fun about RISE, that you can connect with researchers in total different fields, and that gives good ideas grow because of that.
And
so it seems, sorry.
No, just end the sentence.
No, I see that you sharing the screen now of the website at RISE, and I guess you are moving us into the farm office at RISE to make it easy.
How we can come in contact with you and your team and all of the researcher. How do you do that?
Yes, and started there. ri.se is a good start, but you know, we have 3,000 employees, and there is a huge number of web pages at our website. So you better type from office and to get into the focus of drug development, and then we have a kind of site within the site, and here hopefully you could be directed to what you need, and there is this is in Swedish, but there is an English version as well. It's a form that you can fill in and reach us, and we also have could explain just what else we could do, the expertise, maybe the client cases, but the one above that one.
Yes. So I will post the link in the chat. I can do like this because it's now 9 o'clock. Time flies when you have fun. So I want to thank you all for participating this interesting morning. And also, Matilda, I hope it's coming more breakfast seminars.
Yes, let's focus on the end of February, around that time.
And you will find it at Farm Office website or in the LinkedIn or in different ways. So we will come back to you, and I hope you find this interesting and would like to join, and also tell other people that you know to join this seminar. So with that, should we say just from all of us to all of you, we wish you a very Merry Christmas and a Happy New 2023. So thank you all so much for this morning, and have a great day.
Thank you, Cecilia.
Thank you. Thank you. Yeah, get hands. Perfect.